This is the Marine Discovery Centre's research vessel, Penghana. We study the marine environment, measuring the biotic and abiotic parts of the ecosystem. Today we are working near Snake Island in the D'Entrecasteaux Channel. We take a water column profile of the water temperature and salinity. Salinity is the amount of salt in the water. The temperature of the water varies according to season and also with depth. The salinity of the water varies due to influences from freshwater rivers and the amount of rainfall and evaporation. Salt water is more dense than fresh water, and cold water is more dense than warm water. Next, we measure the amount of dissolved oxygen in the water. This is an important indicator of the health of the marine ecosystem. Oxygen can diffuse into the water from the atmosphere, and it is also produced in the water during photosynthesis by marine plants, seaweed and phytoplankton. Cold water can hold more dissolved oxygen than warmer water, and fresh water can hold more oxygen than salt water. We use a Secchi disc to measure the photic depth, or how far the light travels into the water. The point where the disc is no longer visible is called the extinction depth. The photic depth is affected by the turbidity of the water. Turbidity is a measure of suspended solids in the water, which affect the colour of the water. More suspended particles in the water means higher turbidity, and therefore reduced photic depth. Fewer suspended particles in the water means less turbidity, and therefore increased photic depth. A standardised colour scale gives an indication of the kind of suspended particles in the water. This is used in conjunction with the Secchi disc. The pH of the water is measured to monitor ocean acidification. Carbon dioxide dissolves into seawater, forming carbonic acid, and this lowers the pH of the water. Ocean acidification is a problem for animals that biomineralise calcium carbonate, such as corals, animals with shells and exoskeletons, as well as phytoplankton. We use a small benthic dredge to take a sample of the sea floor biodiversity. It is towed along behind the boat through the sediment. We classify, count and record the animals we find, and then they're returned back to the sea. The data we collect on the abiotic and biotic conditions of the ocean help to identify changes and inform how we manage the marine environment.